an introduction presentation about zoonosis. Just before I start, the way we will proceed after this introduction presentation, that we'll talk about systems. We'll talk about each system. We'll talk about the infectious diseases or bacterial diseases as well as viral diseases of each system, basically. And then whenever we talk about zoonotic disease, we will incorporate clinical signs and control measures, biosecurity measures in humans, okay? So if we look for a clear definition of zoonosis, we find more than one definition, but probably the one that presented by WHO in year 1951 still is the more comprehensive one and the most used one, which is diseases and infections which are naturally transmitted between vertebrate or vertebrate animals and man. So I want to concentrate or kind of like grab your attention to vertebrates. It means like, if you are whatever disease you get from animals that are not vertebrate animals, those are not considered zoonoses. But later on, they actually included that or those diseases in the equation. So they start basically considering those as zoonotic diseases. But this is the most commonly used definition, okay? But this definition is still lacking the fact that the origin did not consider the origin of the disease. It's originated in animals or originated in human. So they added more into this in order to give a more clear definition of the terminology zoonosis. Okay, some people they write it with E, like zoonosis with an E or zoonosis with an I. I refer to the latter between the two S's. Okay, and both are correct. So they are not different from each other. So it's like a style of writing, and some people refer like they say this is the English term and this is the American term. I don't care. Zoonosis, whether you write it with the I or E, it's the same, or whether you like zoonotic diseases, it's all the same. So we exclude toxins, diseases that basically where the bacteria just produce the toxins and people take the toxins. This is not a zoonotic disease. We exclude as well diseases that are transported by arthropod, but later on, as I said, they start introducing those like malaria. So the other terminology that I would like you to appreciate are emerging and re-emerging. The word emerging means that the disease did not occur before in a certain geographical area and start basically occurring in that area. So if you have a disease that never been reported in Jordan and suddenly that disease start happening in Jordan, if it's a communicable disease or a zoonotic disease, we refer to it as an emerging zoonosis. An example of that is Rift Valley fever. Rift Valley fever has never been reported in Jordan. So if a case or an outbreak happened in Jordan, then we refer to that as an emerging zoonotic disease or an emerging zoonosis. Now, the word re-emerging means that the disease occurred before but disappeared for a long period of time and start happening again. All right. And for example, in Saudi Arabia, if Rift Valley fever happened again as an outbreak in Saudi Arabia, we refer to it as a re-emerging zoonosis because 20 years ago, Rift Valley fever was reported in Saudi Arabia and resulted in killing a good number of people there. All right. If we look at the map, this is an old map, but I want you to appreciate the dollar numbers here. The cost of whether losses or control of those diseases in different parts of the world. Okay, so if we look at, for example, the E. coli 0157S7, which is a major foodborne disease in North America, which resulted in meat recall and destruction of the meat market in the United States of America in year 2001. It cost to basically, whether it's prevention or direct cost, about $250 million of the federal money, basically. In UK, during the year between 1990 or years between 1990 to 1998, basically, BSE, which is bovine spongiform encephalopathy, اللي هو mad cow disease, costed about $9 billion losses in the beef industry. Why this is a huge number? Because actually Great Britain is a major exporter. So that resulted in crushing down the beef trading for UK. Another example is the avian influenza, the H5N1, all right, in Hong Kong, that resulted in about $22 million losses, and so on, guys. So I need you to basically know what kind of losses we are looking for, and that resulted in basically major crashes of the economy in those countries. So it's not a simple thing. And in addition to the losses that happened in those countries, we experienced even more losses that is a little bit unrelated to the disease directly, but can't be basically taken out from the disease equation, such as the losses in international travelers. People stopped traveling to UK when the BSE outbreak were at its highest peak. Migrant workers basically lost their work, and this is according to the International Labor Organization, منظمة العمل الدولية. Refugees, they basically lost, a lot of people lost their works and they became refugees. Undocumented migrants, migrants victims or trafficking. This is all basically led to more losses as a response of emerging zoonoses. And to understand this in a better way, we need to basically elucidate the reasons for emergence or re-emergence of those zoonoses. The number one reason is the increase in the population of humans, because records have shown that the population of Earth increased tremendously during the last 2 decades. And this increase resulted in huge demand for food, which resulted in providing low quality feed to these people, and to basically providing less quality of other resources such as water, which resulted in more and more diseases and more interaction between the human and animal interfaces. Okay. So and as a result, more trading start happening and less control start basically to be implicated as a result. So in 1950, for example, this is in the these are records from the World Trade Organization. According to that, the international trade of agricultural products in 1950 were about 5 times less than that in 2000, in year 2000. And this actually huge difference or increase in trading resulted in basically loss of the control systems, as those control systems were not able to cope with the huge demands as a result of increase of the world population. Now, the other thing that I would like you to remember, and now the only thing to remember those are to recite them. Some terminologies that you're going to face when you study more and more, and when you do deep dives in the world of zoonoses. The first one is the orthozoonoses. All those orthozoonoses are diseases transmission cycle that can be completed with only one vertebrate reservoir. So orthozoonoses are diseases where the cycle only completed with one vertebrate reservoir, method rabies, such as rabies. Okay, so you still need the dog in order to complete the cycle. So human to human rabies has never been documented. So you still need a vertebrate animal in the middle. Okay. The other one is the cyclozoonosis, where diseases maintain their cycle. To maintain their cycle, they require more than one vertebrate species, such as echinococcosis or teniasis. In case of echinococcosis, you need the livestock, which is mostly the sheep, and the dog in order to maintain the cycle between those animals and the human, right? So the dog eats the cysts and they produce the oocysts in their feces, and those oocysts in the feces are the infected material for a human. A human can't get infection from sheep, they only get infection from dogs. And dogs, they can't get infection from humans, they only get it from sheep. So in order to complete the cycle, you still need two vertebrate species to complete the cycle of the disease. All right. The pherozoonoses, or sometimes referred to as metazoonoses, are diseases that are required to maintain what is required to maintain their cycle, both vertebrate and invertebrate. And we are talking about all arboviruses almost. All right. Such as Rift Valley fever. Rift Valley fever is a pherozoonosis or a metazoonosis. So humans still, they can get the infection from the carcass, but in addition to that, they can get the infection directly from the insect itself or the mosquito. Another example, or a better example, is the West Nile virus or the Japanese encephalitis virus. Okay, where in order for a human to get the infection, they need to be bitten by a mosquito that basically fed on a blood of a horse infected with West Nile virus. The fourth one is the sabrozoonosis, and those are diseases that depend upon inanimate reservoirs or development sites. But they can, the animate ones, they can play a role. And here we are talking about diseases that are transmitted by objects, by feed, by water. Method listeriosis. Listeriosis, usually the animal die inside the silage, all right, or near a drinking water or in the field, and animals fed plants that are contaminated with the carcass or with the dead animal material that resulted in having the Listeria monocytogen entering their body and causing the disease. So the transmission is completed by something that is not life. That's the idea, okay? And that process, we refer to it as saprozoonosis type. Now, for each disease, not just zoonosis or zoonotic diseases, for other diseases, we need to understand the reservoirs, because in order to have a good and a complete way to control or plan to control a certain disease, we need to understand how the disease is maintaining itself. And diseases are maintaining themselves in reservoirs. All right. So reservoirs are or inanimate objects where the infectious agent usually lives. And usually this infectious agent lives in that particular subject, whether it's a human, whether it's a living thing or an object, and maintained from any environmental, basically, destructive factors until they reinfect another animal or a human. All right?
The time is
Sometimes
6:00 p.m.
for a pathogen to infect a human, they need to move from more than one reservoir. And here we call it that the pathogen passed through different reservoirs or did successive passages before reaching the final host, which is the human. A great example of that is the H1N1. Do you remember H1N1 influenza, guys? مين بيقدر يحكي؟ مين بيقول لنا شو ال H1N1؟ معايا انتوا؟
Yes, doctor, the swine flu.
Yes, swine flu. Thank you. Swine flu, what happened that the virus, which is a segmented virus, by the way, a highly segmented virus, so it's the antigenic drift and shift in this virus is very high. So the virus moved from one vessel to another, and usually the vessel was here, the pigs, until they restructure to form a genetic form that can infect humans and cause severe illness, right? Which is the swine flu or the H1N1 disease. Another example here, they are talking about leptospirosis, anthrax, okay, and so on. Okay. So we need to understand what are those reservoirs, what are the sources of those reservoirs, and we need to understand the concept of vectors. And the vectors, they could be biological vectors, and they could be mechanical vectors. And in case of biological vectors, this is where they take the virus, they multiply it in their body, they even may pass it to the eggs and eventually produce another progeny or another, basically, vector that carries the same virus. And it could be not even the same virus, it could be a modified, genetically modified virus, or a virus with a mutation that can cause more harm to a human being, okay? So vectors are a living organism, frequently arthropods, insects, that communicates an infectious agent from an infected to a susceptible animal or a human being. For example, the yellow fever, it's the major vector of that is the Aedes aegypti. Okay? الزاعجة بالعربي الزاعجة المصرية يعني. Aedes aegypti. This is a mosquito, guys. All right. Okay. So this vector could be again a mechanical vector, or this vector could be a biological vector, and it depends whether they take that pathogen and basically have that pathogen multiplying in this body and pass to its progeny or not. That's how you differentiate whether it's a biological vector or a mechanical vector. طيب في أسئلة لهون شباب؟
دكتور أنا عندي سؤال.
معلش هاي فيها terminologies كثيرة لكن please spend half hour to remember those. لأنه أنت لما تقرأ in the future مرات أنت بتضطر إنك تسمع كلمتين بالأخبار a documentary in English about zoonosis. So you are required to know zoonotic diseases more than the human doctors. You know what I'm saying? So please try to read this. If you have questions, come back to me and we'll discuss it more. I have to add one thing before you question. في عنا احنا حتى according to the origin, as I said, the WHO in 1950, when they decided to give that definition of zoonosis, they actually did not mention whether it's originated in animals or humans. And later on, a lot of scientists start talking about this. And then they said, well, okay, diseases that are originated in humans and transmitted to animals, and diseases that are originated in animals and transmitted in humans. So I want you to remember those two basically terminologies. I'm gonna write it in the comments section here. Where is the comments? I have to terminate this in order to reach the comments. Yeah, here. Just 1 minute, guys, because I'm messing up things here. Where are the comments? Chat. In the chat box, please. So we have the anthropozoonosis. This is the first one, anthropozoonosis, okay? And the second one is the second one is the zoonosis. Take those two words and see which one means it's originated in humans and transmitted to animals, and which one is originated in animals transmitted to humans. I think zoo is two O's here, but I'm not sure about the second wording. The second word here, please check. Zoobozoonosis. Yeah, zoobozoonosis. Zoobozoonosis. Like this. Yeah, what is your question? Yeah, first, okay. Let me see how many hands we have. We have more than one hand here. Let me answer first. Zed.
Yes.
Go ahead, Zed.
In the first slide that talked about the definition of zoonosis, it was they mentioned the experimentally transmitted diseases. What do they mean by that? Experimentally transmitted diseases.
Experimentally transmitted diseases are diseases that are basically set in the laboratory, which means that intentionally did the infection. Okay?
Okay, thank you.
Who else? There was a girl. مين البنت اللي كانت بدها تحكي بالله؟ انا اسف
دكتور أنا كنت بدي
سكتتها. مين؟
أنا كنت بدي أسأل
اسال مين انت؟
لما إحسان
مش ظاهرة عندي. ايش اسمك؟
إحسان
يا احسان تفضلي.
دكتور كنت بدي أسأل لما حكينا عن ريسيفير هلا الباسج اللي بيصير يحدث بسبب إنه الزونوتيك ديزيز مش لاقي فاينل هوست ولا لأنه بده يعمل تعديل على نفسه على حد ما يجد الفاينل هوست؟
لما حكينا عن ايش؟
الريسيفيرز
ريزرفوارز. الريزرفوارز
بسلايد 9 دكتور.
الريزرفوارز قصدك؟ سلايد 9.
أعتقد
انا ارجع سلايد معلش. لا مش هاي. سلايد 9 هاي الريزرفوارز او الريزرفوار بالفرنسي، ريزرفوارز بالانجليزي. It could be like this. It could be like this and it could be that the virus is hibernating. It means انه بده ينام. يعني يعني كلمة ينام يمكن الناس تضحك عليها، يقول لك شو فيروس هذا بده ينام. But really viruses sometimes they hibernate. They stay over winter until they their major vector number, the population of the vectors that transmit the virus comes to life, and then they start infecting again. So a reservoir is where the virus basically kind of like maintain itself. It could replicate as well and get amplified to more numbers basically. And sometimes they do go into genetic alterations to become a more virulent strains. So it could be just a station where they stop until the vector or until the opportunity to be in another host happening, okay? Or they stop there, they start multiplying, or they stop there, they start multiplying, and they start producing a new genetically altered virus, okay? So it's a station. Look at it as a station where the virus stays. It could be to protect itself, and it could be to search for another host, and it could be to basically get ready for the other season, the second season, where its transmission tool is available.
شكرا دكتور.
مفهوم؟
تمام.
Good. Okay. طيب. بعد ما خلصنا هاي الانترودكشن about zoonotic diseases بدنا نبلش نحكي عن السيستمز ودiscuss السيستمز. اول سيستم رح نحكي عنه اللي هو المامري جلاند بالنسبه للانفكشس ديزيزز، واول شيء رح نحكي عنه اللي هو البوفان ماستايتس. شباب شايفين السلايدز؟
دكتور ظاهر.
طيب ايش الماستايتس؟ مين بيقدر يعرف لي شو الماستايتس؟
هذا الانفلاميشن دكتور.
inflammation of the mammary gland. مش هيك احسن؟
دكتور.
inflammation of the udder دكتور.
طيب شو يعني inflammation؟ من الاميونولوجي كلاس اللي انتم درستوه، انتم ماخذين اميونولوجي صح؟
دكتور.
So what is inflammation? Who can define it?
دكتور
Yes.
بتقدر تنزل الشابتر على الاي ليرنينج؟
انت سيف احنا قلنا بننزل لك كل الماده تاعت الميد تيرم على الليرنينج ماشي؟
بس متى دكتور؟
ASAP. ليه؟ يعني احتمال اذا مش اليوم بكره يعني هاليومين ننزلهم كليات ان شاء الله لحد الميد تيرم. شباب شو يعني انفلاميشن؟ يلا فوتوا على جوجل.
دكتور بصير في عندنا انكريز بالفاسكولار permeability
ايوه
بسبب افراز الهستامين،
حلو.
وبصير في عندنا redness
as a result of what؟
افراز الهستامين من الماكروفاج.
هيستامين لوجه تفرز من
وجود أفراز
الماكروفاجز. ها شباب don't go into deep things. Think simple. Inflammation is a response, right? مظبوط. Inflammation
Yes.
is a response to what?
Offense دكتور.
infection.
Yeah, an infection or what? Or a damage that happened to a certain tissue. So the body response to an infection or a damage that happened to any tissue, the body responds by inflammation. All right. So first thing happened that regardless what kind of hormones, what kind of mechanism, how it happens, but بيزيد الفاسكولاريزيشن اول شيء. فبصير في عندنا migration للانفلاماتوري cells اللي هي الوايت blood cells to the location where that damage or infection happened. Okay. As a result بصير عندنا اللي هم الكاردينال ساينز تاعت الانفلاميشن. شو الكاردينال ساينز تاعت الانفلاميشن؟ شو هن يعني العلامات الرئيسيه انه هذا الجزء من الجسم inflamed؟ شو هن؟
pain, redness, swelling,
ايوه
swelling,
pain
heat,
pain redness. شو كمان؟
swelling,
swelling
loss of
ماشي.
function.
loss of
Lose
function.
the function.
يا سلام عليك، ممتاز يا حسام، احسنتوا يا شباب. اذا mastitis is تعرفوا انتوا الايتس معناته انفلاميشن، مظبوط؟ من البيسك ثينج اور البيسك كونسبت بتاعت اللي تعلمتوها انا متاكد انه بالامراض انه كلمه ايتس اذا حطيتها كولايتس معناته انفلاميشن اوف ذا ايش؟ of the gallbladder. If you put hepatitis, inflammation of the liver, right? If you put like whatever, right? Itis or titis, huh? So in this case, mastitis, inflammation of the mast, right? Which is the other, ماشي? So inflammation of one or more of the quarters of the other. And here, as you see, an inflamed udder will basically show the four things that your friend talked about: swelling, pain, warmness, and redness. All right. So, how important mastitis is? It is significantly important. Why? Because if you take the milk from a cow, then you are really reducing its value to less than 1/4. Because a dairy cow, a lactating cow, worth the amount of milk it will produce during the production cycle. All right. So usually a cow produces about $200. Sorry, much more than $200. But the losses due to mastitis is estimated to be about $200 per cow per year, and this is in the United States of America, guys. This is in the US. So if mastitis causes about $200 losses per cow per year in America, then just imagine how much it costs of losses in our countries. Okay. We all know how good the medical system, the feeding system, the management system in the States, and still it causes about $200 per cow per year. All right. So it is estimated that in the whole states, United States, this disease, mastitis, cause about $2 billion losses per year in milk. And this is due to what? Due to treatment costs, due to culling, because mastitic cows, if they don't respond to treatment, they are culled. All right. Culled means getting rid of from the herd, or they die. RIP means rest in peace, yani dead cows. Okay. So the major or the number one cause of losses in dairy industry is not FMD because of FMD. It's because FMD causes huge reduction in milk. But the reduction in milk due to FMD is temporary. But the reduction in milk due to mastitis, especially if it's a chronic mastitis, is permanent and usually results in killing those cows. So what are the concerns that we have due to mastitis? When we look at cows, the concerns is the loss of functions of the quarters, the reduction or steep reduction in milk yield, and loss of the animal, death of the cow. And if we look at the human health, yes, most of the mastitis, they don't cause or they don't basically cause any disease to human, the mastitis pathogens, most of them. But if a cow got mastitis, then they're gonna produce low quality milk. So we will have low or poor quality milk. And then if there are no controls or regulations related to treatments, then you will be getting a lot of milk that is full of what, guys? Antibiotics. And this is even more dangerous because this could result in what we call AMR, antimicrobial resistant bacteria, to be emerging and to cause more troubles, such as اللي حكينا عنها اول وحده الايكولاي 0157S7 اللي هي known to be a multi-drug resistant bacteria, MDR. Multi-drug resistant bacteria, we refer to them as MDRs. Okay? Now, how severe can mastitis be? Mastitis can be really severe. And when we talk about the clinical picture of the mastitis, whether clinical signs are appearing or not, we can classify mastitis into two types or two classifications: subclinical mastitis or clinical mastitis. Let's talk first about the clinical mastitis, which is a lot of people think that clinical mastitis is more severe than subclinical mastitis. As a matter of fact, clinical mastitis is easier to handle than subclinical mastitis because you see it. هسه اهم شيء بالمشكله عشان تجد حل لاي مشكله انك to see it. مظبوط. So clinical mastitis, you see it, it happens in about 5 to 10%. It causes inflamed udder. We see changes in the milk, clumps and clots in the milks. Okay. In acute mastitis, acute clinical mastitis, major types of clinical mastitis are acute. Bad milk, loss of appetite, depression, and prompt attention is needed to these animals. Otherwise, we're going to lose them because they may die. In chronic mastitis, you get the bad milk and cow appears healthy. However, it doesn't milk and tissues became fibrogenic and more like, hardness happening in the udder, and most of the time cows lose the capacity to produce milk in case of chronic clinical mastitis. Now, In case of subclinical mastitis, this constitutes about 95% of mastitis cases. So the majority of cases that you're going to see in the field of mastitis are subclinical mastitis. The other appears normal. You can't tell whether it's abnormal or not, but you get milk that appears normal. However, you get steep reduction in milk yield, right? And you get an elevated what we call somatic cell counts. What are those somatic cell counts, guys? شو هن السوماتيك؟ شو معنى لما احكي somatic cell counts? What do I mean?
Antibodies present in milk.
ايش؟ antibodies؟
دكتور antibodies present in milk.
لا، مين بيعرف؟ اقول ما درستوا بالهيستولوجي ايش يعني somatic cells؟
الخلايا الجسمية.
الخلايا الجسمية. Yes, يعني we could say this, لكن هي in general the best definition are cells that contain nucleus. Somatic cells are cells that contain the nucleus. يعني can we say انه red blood cell is it a somatic cell?
No.
طيب neutrophils
بلى.
right. الابيثيليال cells
بلى هدول اللي فياتهم نوكليوس.
yes. إذن أي cell جيدة هي. أي cell contains a nucleus is what? Is a somatic cell. So increase in somatic cell counts. معلش أنا شلت السلايد لأنه بدي أشوف مين اللي بيحكي معي. Increase in somatic cell counts means that there's inflammation happening. Either there's destruction of the epithelial cells of the other, so you get more epithelial cells, or due to inflammation you have more white blood cells coming to the udder and mixed with milk, and this is where you get the neutrophils mainly. All right, and those are the main constituents. The epithelial cells and the neutrophils are the main constituents of a subclinically infected milk, and it takes longer to treat. So it's more severe actually to have, or more economically important to have a subclinical mastitis than a clinical mastitis. What causes mastitis? 70% of the mastitic cases are caused by bacteria. 2% by yeasts and molds. And this is where you guys, when you go to the clinic إن شاء الله بعدين راح تشوفوا إنه الكاو مبين إنها infected، والأونر بقول لك أنا ما أعطيتها أنتيبيوتيك، ومع ذلك بناخذ عينات حليب بنبعتها للمختبر ما بيطلع أي شيء. ساعتها بنحكي إحنا للمختبر please check for fungus, and this is where it hits. Okay, and we have a lot of unknowns, actually about 28% unknowns due to trauma or due to any physical alterations rather than bacterial, and those are treated after you remove the source. So if it's a trauma, you fix the trauma. If it's a cold weather, if the cow is kept outside during a severe weather, you ask the owner to bring them in the warmness, and usually they start recovering directly. All right. So where do these organisms comes from? Usually they come from the environment, and that's when we refer, or we have, this is where we have another classification of mastitis. We said we have clinical, subclinical, right? But now, according to the source, we have two major types: the environmental mastitis and the contagious mastitis. Environmental mastitis is the mastitis that is caused by pathogens that are found in the environment of the cow. Most of the time, the bedding of that cow. All right? The most important pathogen of those is the what? E. coli. E. coli is number one. We have some types of Klebsiella pneumoniae, and we have others as well, majorly the coagulase negative, the non-coagulase negative Staphylococcus. Some of them, basically, they all Pseudomonas as well. They all referred to as environmental mastitis. The other type is the contagious mastitis, and the contagious mastitis, guys, probably the most important is the of contagious mastitis. The most important thing to understand about contagious mastitis is that the mastitis is transmitted by equipments or by the hand of the milkers. And most of the time, the equipments are the transmitters or the inanimate vectors or objects that transmit the bacteria to the healthy udder. The most important pathogen known is, and the most important one actually, is the Staphylococcus aureus. In addition to that, Staphylococcus agalactia as well, Streptococcus microorganisms, other types. And the most important of the streptococci is the Streptococcus agalactia. Now, if we look at pathogens, streptococci that cause contagious mastitis is the Streptococcus agalactia. However, other types, herpes, or dysagalaxia, or equins, they are environmental and they cause environmental mastitis. Okay? And usually we see clinical mastitis in case of contagious mastitis, and most of the time we see subclinical mastitis in case of environmental mastitis. And even the way we treat environmental and contagious is a little bit different, and the way we control or we try to prevent is a little bit different as well. Okay? In the field, لما بتروح لامريكا اوروبا كده، لكن مش بالفيلد تاعنا. Strips means streptococci. Environmentals means the pathogens or the bacteria that type. Staphylococcus, we have the most important one is Staphylococcus aureus, and some people refer to it as the summer mastitis. And it is a contagious mastitis, basically spread by milking equipments and milker's hand, and it persists in the udder for a long period of time. And most of the time, they are very difficult, or they can't be eliminated. They can't be treated. And there is an expression in America. We used to hear it when we go to the field, that once a staph cow is always a staph cow. Okay, it means that you can't treat a staph cow. And even most of the time they refer to it as a staph mastitis. Other types of staph that usually are found and they increase the somatic cell counts, they are more an environmental, as I said earlier, than a contagious ones. And they respond to antibiotics and they are treated, but they are oftenly relapse, because relapse infections. Coliform mastitis, those are environmental mastitis. We call it coliform because of the nature of these organisms produced a coliform bacteria. Those are the three major bacteria within this group: the E. coli, the Klebsiella, and the Enterobacter. And they are environmental, and they are usually preserved in the manure, in the bedding, okay? And they cause high temperatures, inflamed quarters, watery milk with clots and pus and toxemia. All right, and usually there is a vaccine for that. We refer to it as J5 vaccine. The J5 vaccine is one of the first vaccines produced to treat mastitis. Actually, it contains lipopolysaccharides, which is the endotoxin of the E. coli. And I was one of the people that developed this vaccine, the J5 vaccine, when I worked for Merial in the year 1998, I think. Most of you كنتوا مولودين ب 98 يا شباب. انتوا الدفعه مولودين بال 98 ولا لا؟ الماجوريتي انتوا مواليد اي سنه؟ 2000؟ لا شو 2000؟ 2001 انتوا صح؟
2002 دكتور.
2002. يعني هذا الفاكسين اشتغلت عليه انا قبل ما تنولدوا ب4 سنوات. يعني خلاص بدي اوقف لانه هذا معناته الواحد ختير. اوكي طيب. Other organisms that are important سيدوموناس زي ما قلنا، الجونوزا وهي environmental mastitis caused clinical mastitis. Seriatia not very important, but other important ones is the Corynebacterium بايوجنز، واللي هي بتعمل عندنا الكورينبكتيريم بايوجنز والاكتينوبايوجنز. هذول very important لانهم they produce small abscesses and they cause abscessation of the adult lymph nodes. Fungi, candida قلنا هذول represent 2%. And the newcomer in the field of bovine mastitis is the Mycoplasma bovis. Okay, Mycoplasma mastitis, and this is usually a secondary to another infection. So usually a calf or a cow, sorry, with Mycoplasma bovis mastitis, usually that cow will suffer another disease or another syndrome, which is pneumonia. Okay. So how mastitis happened? So in order for mastitis to happen, you need different predisposing factors to be present. As I said earlier, an inflammation is a response of a trauma or a damage to the tissue. It's either by a pathogen, an infection, or by physical damage, right? So you need initially something to disrupt the nature of the epithelium of the udder, especially the udder canal or the teeth canal. Okay, and the most important or the most important damage source that is has been studied and documented is the milking machine itself, because a lot of people, they don't maintain their milking machine pressure as well as the cups in well health manner. So they may have a pressure that can produce some kind of trauma to the epithelial of the canal of the teeth. All right, and this actually particular disruption, we refer to it as the back pressure. This is how we refer to it, back pressure. And this is when you have very strong unestimated type of pressure and tuned, and then when you stop that pressure suddenly, what happens after stopping the suction, there will be strong let's say reflex that will happen in the other side or the other end of the pressure, and this lead to what we call a backflush, which means that the orientation of the epithelial cells during suction will be downward, and when you stop that suction suddenly, and if the pressure that you used is not the correct pressure, you will get a backflush of this air that has been sucked, moving those epithelia towards the opposite side, the opposite end. So instead of being oriented downward, they will be basically pushed quickly upwards, and this will cause a certain type of destruction of these epithelial that will eventually allow pathogens that are opportunistic pathogens to go inside and cause the inflammation. Other things that could cause mastitis are teeth injuries due to diseases or due to physical, basically, causes, such as cows when they stand up, if they have Bandolius udder, they may step on the teeth, okay, and sometimes they fight. Cows, they fight intentionally or unintentionally. They hit each other's teeth with their legs, causing damages. وهذول كلهم بيساعدوا انه يصير في عندنا opportunistic bacteria to get inside the udder and cause severe problems. During sometimes some cows that are weak and not fed well, they will have low immunity that lead basically to opportunistic bacteria to work, and most of the time the lowered immunity may result after calving or after a certain type of surgery where you give anti-inflammatories. Okay, so the nutrition, as I said, play a great role. So the other important player is the organism. What type of organism there? What type of toxins they produce? What type of pathogenesis they basically use in order to cause the damage to the tissue? And thirdly, the environment. So if you have an environment that can maintain the pathogen for longer period of times, so if that environment is always humid, is always warm, hot, humid, and if the owner of those cows, they don't clean regularly under these cows, they will result an environment that is full of opportunistic bacteria that is just waiting to get inside and cause problems, because cows eventually will lay down, right? They will sit, they will lay down, and when they lay down, if they have leaky udders or if they have some kind of wrongfully oriented teats, okay, they will allow bacteria to go inside those canals and eventually reach the mammary tissue and cause the problem. So this is what I just said. So they invade through the teat canal, they migrate up until they reach the tissue and eventually produce their toxins and cause the inflammation. And the immune system responds by sending somatic cells, which are the white blood cells mainly, neutrophils. So the outcome of the inflammation, it's either will clear the infection and animals will recover, or they will become clinically infected or subclinically infected. So the challenge is how to diagnose subclinically infected cows, because in clinically infected cows, easily you can just basically know that they have mastitis by looking at the type of milk and by observing the different cardinal signs of inflammation. But in case of subclinically infected cows, physical examination may not help. So then you need either to do somatic cell counts or to use a different bin side or cow side tests, such as the California mastitis test. مين بيعرف هذا؟ مين عمره شاف الكاليفورنيا mastitis test CMT؟ حد منكم عمره اشتغل بالسي ام تي؟ عمركم سمعتوا فيه السي ام تي؟ شباب معاي ولا روحتوا؟
لا دكتور أول مرة نسمع فيه.
طيب CMT is a chemical test. شايفين البادل هاي فيها أربعة ويلز كبار. What you do, you milk from each teat, from each udder, in those four wells, and you add equal quantity of the CMT chemical. The CMT chemical is a chemical that basically interacts with the DNA. So this is a chemical that interacts with DNA, and the interaction results in formation of a gel. So, and usually we give it's colored. It's there's a stain there, methylene blue. So it's a blue color. So when you add the milk and then you add the CMT and you start swirling, those that have CMTs as somatic cell counts will have a lot of DNA, and then they will result in forming a gelatinous material inside the cup. And those that don't have somatic cell counts will stay liquidy, okay, without formation of any gelatinous material. That's how you know. And the other thing is to send samples to the laboratory, and this is the best thing. But it is a very costly procedure. Now, treatment, usually we always, unfortunately, go back or always tends to concentrate on giving antibiotics. And most of the time, those antibiotics are given without any studied decisions. All right? The rule of thumb is that you should test what type of bacteria you have and what type of antibiotic is the best choice for this bacteria by doing culture and sensitivity testing. The other thing, sometimes in case of clinical mastitis, we need to clear the infected milk from the udder. So it's very important to clean the udder from whatever infected milk in it. And usually we give oxytocin, which will promote the production of more milk. So as you produce more milk and you clean it and you empty the udder, you are creating a flushing effect of whatever pathogens and infected materials are inside the udder. Okay? So always, every 2 hours, remove the milk from the udder. So don't keep the media, because the milk is the best media for bacteria. So don't keep the best environment for the bacteria to nourish and to increase in number. And then you treat with antibiotics. And you treat with antibiotics based on sensitivity testing, not always penicillins, as it's written here. For subclinical mastitis, treatment is questionable because actually you really don't know, and you always come to realize that you have mastitis late. So if you don't examine your cows on a regular basis by somatic cell counts and by California mastitis test, and this is something that most people they don't do, they are late in treatment. And even when you pick it up, when you diagnose it and you are sure that you have subclinical mastitis, it takes long period of time to clean. All right? And usually it takes 1 month to treat a subclinically infected cow. So you will be losing milk from that cow for 1 month. So this is the huge loss that you need to suffer if you have subclinical mastitis. So the best thing to treat subclinical mastitis is by preventing subclinical mastitis from happening. And here I'm mentioning 10 steps for mastitis control. I don't want to go through these steps. I want you to read them, but the steps basically concentrate on biosecurity. And biosecurity depends on 3 major things: cleaning, disinfection, and restrictions. All right? So first of all, you need to clean the teat, the udder and the teat before milking. So we have special materials we call breed depths. Breed depths materials, those are the materials that is used before you start milking cows. Okay? Using of breed depths, research have shown that using of breed depths reduced infection of the udder by 70%. So this is even much better than using antibiotics. We have the iodophores. This is the most widely used, but chlorhexidine is used, but it's very expensive. quaternary ammonium materials is used and other materials are used, but we need to be careful not to use materials that cause stress to the other tissue from the outside, like hypochlorous acids or bleach. Then afterwards, you clean these disinfectants by special towels. You dry before you put the machine into action. When you put the machine, you have to properly test your machine. Make sure that the pressure is correct. Make sure that you are not over pressuring the teeth. Make sure that you don't have back flushes, as I said. Make sure that you milk the cow for a certain period of time, not to over milk the cows, because when there's no milk and still you're sucking, it means you are damaging the other tissue. All right? And then make sure that you are cleaning, that your machine is cleaned itself. All right, and you apply it and remove it carefully. And dipping after each milking is very important. And the dip that we use after milking, we call it post dips, and those actually should seal the teeth canal. So it's a special material. There's a special material in those chemicals. They are similar to the pre-dip, but in addition to that, they have special material that seals the tip of the canal. So they are not allowing bacteria to go inside for at least 8 hours, 6 to 8 hours. All right? And then you need a special monitoring system. DHI is the Dairy Health Improvement System of the somatic cell counts. This is for North America. We have another system for Wisconsin. We call Wisconsin mastitis test. All right. Those are the processes. I wish that we have some kind of networking here in Jordan to apply. Clinical cows should be treated and milked at the end. Always chronically infected cows should be milked last, and those should be in the list of culling. All right. Drying off or dry cow treatment is quite important. I will be giving you a special presentation of dry cow therapy. Please read it. This is a self-reading material. You read it on your own. Come back to me if you have questions about dry cow therapy, and I always ask questions about dry cow therapy. All right. And this is a very good technique to lower the risk of clinical and subclinical mastitis during subsequent lactations. Keep cows clean and other free from soil and manure. So this is very important. And always boost the immunity by giving proper feed. So mastitis is primarily a management problem. Mastitis can be controlled, and prevention programs work best and better than giving any antibiotics. And I've included some procedures. This is for your reading, basically. I will not be asking any questions about from this slide forward. I'm not going to ask you any questions because this is more a kind of managemental rather than disease-oriented materials. But this is one thing that can be used in order to basically prevent further problems related to mastitis. The HACCP system, I'm not going to ask you about HACCP as well, so you just go ahead and look at it. Do you have questions, guys? معاي ولا نمتوا؟
دكتور أنا عندي سؤال.
تفضلي.
بالسي ام تي
تفضلي.
بالسي ام تي بس بدي أعرف الجيلاتين فورميشن بعمل لي كأنه بوزيتيف، يعني في عندي
gelatinous formation of gelatinous material. Let me just معلش انا قاعد بدور على تيكت هون، بدي اشتري تيكت عشان اروح. طيب يعني لو جينا عملنا هون CMT test mastitis cows You see this? The picture. Let me see videos. أنا 2-minute فيديو. شايفين معي الفيديو ولا لا؟ شباب شايفين معي الفيديو ولا لا؟
دكتور بس لسه بحمل بدور.
قاعد بحمل انا لاني ساكن باوتيل المفروض يكون اسرع من هيك. طيب هيا شايفين كيف حط equal amount of milk في
Okay, well, we're
البادل.
going to perform a California mastitis test on this cow. That's going to test her to see if she's got subclinical mastitis, and it'll look for any quarters that are about 400,000 or more in terms of cell count. So it's useful in herds that don't milk record. What we're going to do is use the paddle, which has got 4 little wells. We can collect a milk sample from each quarter into that, and then we need some of the reagent handy, which is a detergent type reagent. So the first thing we'll do is get some milk from each one of the quarters.
يعني equal amount of milk in each
so we have the milk
well,
from each quarter of the cow. We know which one they respond to which quarter because we watched when we were doing it and made sure from the direction we've done it that we can record which one's which. We can then tilt the paddle over. So
لاحظوا كيف عملت لها tilting.
Which was shown by one of these lines marking the graduation.
شايف كيف عملت لها لسه ما she did not add the reagent.
And then we get the reagent, add the same and equal volume as the reagent as the milk. So we swirl this round, and what you're doing is looking for a gel type reaction. You can see that in here, these have all gelled up, whereas this one looks more milky and is easier to swirl around the paddle. So these 3 are going to show up positive reactions, which means they have a cell count in the region of 400,000 or higher. And this can form the basis of either our treatment or our sampling that we might want to go ahead and do for the cow.
اوكي شفتوا الفيديو كيف؟
دكتور
very simple. Actually it's very inexpensive and you can find it in the market في الأردن. موجود بالسوق. في حدا عنده زيد؟
دكتور بس بدي ارجع اسالك اذا صار في عندنا جيلاتين فورميشن معناها هذا الاشي ماستايتس بوزيتيف او نيجاتيف؟
formation of gelatinous material it means زي ما شفت الفيديو شو حكت هاي البيطرية حكت formation of gelatinous material means that there is more than 400,000 somatic cell per 1 ml of milk.
يعني بوزيتيف.
هلا امالك شوي علي.
تمام.
هلا في عندنا عشان انت فوتينا بالحيط لانه لازم سؤالك لازم اشرح لك. في عندنا ستاندردز عالميه. ستاندردز عالميه هاي لكل بلد لها ستاندردز. في امريكا above 250 somatic cell count per 1 ml means that this cow is mastitis والفاكتوريز مصانع الحليب ترفض الملك. ماشي البالك ميلك بنحكي عن البالك ميلك. بتعرف شو يعني كلمه بالك ميلك؟ اللي هو لما يجمعوا من كل الابقار ويحطوها بخزان واحد، هذا البالك ميلك. اذا كان السوماتيك سيل كاونت تاعه more than 250,000 بيرميل برفضوا الحليب. بس بدي احكي لكم شغله، عندنا بالاردن غير مطبق هذا السيستم، لكن احنا عملنا على بالك اللي بروح للمزارع، اللي بروح سوري اللي بروح المصانع. عندنا بالاردن بقدر اقول لك لحد 500,000 امور تمام. يعني انا شخصيا بكون هابي اذا لقيت فقط 500,000 بيرميل. فكل دوله لها يعني تطبق سيستم مختلف عن الثانيه. اوكي. فهلا الجيلاتينس ماتيريالز appears after 400,000 cells per ml. 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 400,000 cel يعني بعد ال 400 بعد ال 350 400 بتصير تشوف الجيلاتينس ماتيريال هاي. اوكي
تمام، شكرا.
عشان هيك بامريكا ما بيعتمدوا السي ام تي، مش معتمد كثير عندهم، معتمد بس مش هو الانديكيتور. عندهم ماشين ثانيه بسموها سوماتيك سيل كاونتر. هاي سوماتيك سيل كاونتر عباره عن يعني زي فلو سايتوميتر بكل المصانع موجوده. they digitally count using the laser power. يعني بمشوا الحليب من خلال they read it through a laser, how many cells are there and it's recorded basically. طبعا في المصانع عندنا المزارع الكبيره، المصانع عندها هذا بسموه الكولتر كاونتر او سوماتيك سيل كاونتر. اوكي حد عنده اسئله؟ طيب شباب، انا ان شاء الله يعني اليوم ما اظنش، اما ان شاء الله بكره بلكي نزلت لكم كل الماتيريالز. اوكي and then if you don't have any questions I'll leave you now and we'll meet next week same time. okay في اسئله؟ يا اما انكم فاتحين ومسكرين يا اما هسه بكتشف. في اسئله شباب؟
دكتور
دكتور
هسه لو يعني لقدام مش ممكن مثلا ست تشرح لنا مثلا بالعربي، يعني لو اخف شوي انجليزي، لانه صراحه اليوم ضعنا شوي.
اكيد ان شاء الله.
يعني انه كثير انت
The
يعني
time
حكيت
is
على كثير بالانجليزي
7
فاليوم
o'clock
ضعنا.
p.m.
طيب ماشي ان شاء الله خير. اوكي
شكرا لك.
طيب guys يلا take care, enjoy the rest of the evening and good night.